And thanks everyone for joining us today. It is my immense pleasure to introduce our speaker for today's seminar, Dr. Madeleine Durkee. Dr. Durkee is a biomedical engineer, and she has been working in the Department of Radiology at the University of Chicago as a postdoctoral fellow since 2018. She earned her doctorate at the Texas A&M University, and she developed and implemented radiative transport models that were aimed at improving the design of optical systems. Ultimately, this work led to enabling the construction of handheld confocal microscopes. At Chicago, she currently focuses on answering questions about spatial biology and human disease, and her postdoctoral work combines computer vision and machine learning and is the basis for setting her eyes on developing methods that can both detect and quantify disease signatures in medical and biological microscopy images. In addition to being a remarkably creative engineer with a passion for engaging students in imaginative ways, Dr. Durkee used to be a competitive figure skater and is very excited about the prospect of winter sports in the Denver graded area. She has built in time for questions during today's seminar, so we all may take advantage of the spaces. Dr. Durkee, we are delighted to have you with us today. The floor is yours. Thank you.
Great, thank you so much for that introduction. So I will be talking to you all today about something that I call deep spatial phenotyping, specifically within some multiplex microscopy images and even super multiplex microscopy images. That being said, I do have a few points within the presentation because I know these hour-long seminars can get long via Zoom, so feel free to jump in and either raise your hand or drop something in the chat, and I'll try to address those questions at those points in the talk. With that being said, oh, now my clicker isn't working. There it goes. Okay, so I'll go ahead and jump right in. So what is spatial phenotyping? So normally when you think about phenotyping a cell, you think of using techniques like flow cytometry and single cell RNA sequencing. So you get very specific information about individual cells. You can get thousands of transcripts per cell, but this requires tissue dissociation. And the spatial layout of cells in human disease can be important for learning about associations with patient outcomes or therapeutic response. So usually people will also look at microscopy to study disease to get a sense for the spatial layout of cells in various diseases. And so fluorescence microscopy allows you to maintain the spatial relationship between cells. However, limited markers can be probed at once, and if we're looking at a fluorescence confocal microscope, top of the line microscopes are limited to about 6 to 7 markers per sample. So definitely not the type of phenotypic resolution that we're looking at with something like single cell RNA sequencing, but we do get that extra dimension of data associated with this. Oops. So spatial phenotyping refers to the characterization and quantification of cells in these multiplexed images. So now if we're thinking about analyzing those multiplexed images, we have to think about how we can pull that quantifiable data out of it. And so we're talking about images, we're therefore talking about computer vision algorithms. So all these computer algorithms are task specific. So if you're talking about a single object per class, there are a few tasks you can do. First is just classification and localization. As you can see in this image here, we have an image of sheep. Very easy to identify as a person. You can localize the sheep within the image, and we can do pixel level classification, which is also known as semantic segmentation. And so you see that every single pixel is either classified as road, grass, or sheep here. But what I do want you to take note of is that as a person, you can see that there are 3 sheep. However, all the sheep pixels are in one bin, so you cannot extract information about the individual sheep in these images. Got to get used to my clicker again. And so then there's multiple objects per class. So these computer vision tasks are also object detection. So I can just draw a bounding box around each sheep with my object detection algorithms. But I can also combine these object detection algorithms with semantic segmentation for a task called instant segmentation, where I can get pixel specific, shape specific information on each object within a class. So if I wanted to compare the features of all 3 sheep in this image, I would have to use instant segmentation to pull off object-specific information. So now if we move back to microscopy, what do we need out of these images? So we need cell class and protein expression and the class frequency data. Those are, that's information that we could pull off out of like a non-spatial technique, but we also want to do spatial mapping. So we want to pull off the cell location, cell shape, and other morphological features. So how do we pull that out of an image? There's conventional image analytic techniques like thresholding, watershed. A UNet is a deep convolutional neural network that can do semantic segmentation, but these all lack accurate object level information. And so even after separating a semantically segmented image, we're not very accurate about the features associated with these cells using these methods. So what we want to do is combine the three tasks of object detection, object classification, and semantic segmentation to get our instant segmentation task, just like that sheep image that I showed you in the previous slide. So how do we do that? So we have state-of-the-art instant segmentation network architecture. So we use deep convolutional neural networks, also known as DCNNs. And so the best one right now for doing that is Mask R-CNN. And so this is a big network, and just to briefly go through it, on the left-hand side, you have your DCNN backbone, which we use a ResNet 101 network with the classification layers stripped off. And then we kind of merge that into a feature pyramid network and build up multi-layer feature maps as you move over to the left. And so rather than doing object detection on the image, you do object detection on those convolved feature maps. And so you have your region proposal network that feeds into those feature maps, and then you pull off those objects at the object level that you will then do semantic segmentation and classification. And then as you're training this network, you do back propagation across all of these layers, every segment of this network, to get all three tasks in one shot. So our first clinical motivation is lupus nephritis. Lupus nephritis is an autoimmune disease characterized by chronic inflammation of the kidney. It is a manifestation of systemic lupus erythematosus. It's characterized by infiltrating immune cells, which can lead to kidney fibrosis and ultimately failure. I'll refer to this as end-stage renal disease, or ESRD. And so these patients will either need dialysis or a kidney transplant if they progress to ESRD. So lupus is a very heterogeneous disease, and there's conflicting opinions on which cells are associated with poor outcomes and the mechanisms associated with that as well. So if we look at this patient cohort that we have collected, you have just a baseline quantification that a pathologist has done of the number of infiltrating immune cells within each of these samples. So patients are characterized by their TI score. And so 0 to 1 is no to mild inflammation, and 2 to 3 are moderate to severe inflammation. If we look at the Kaplan-Meier curve on the right, you can see that if you have minimum to mild inflammation, the patient is most likely not going to progress to renal failure. But if we look at the patients that have moderate to severe inflammation, only half of them move to renal failure. So just the baseline levels of inflammation are not enough to tell if a patient is going to progress to kidney failure or not. So simple quantification of infiltrates is not enough, and we want to know if we can detect outcome-associated differences in inflammatory states through spatial phenotyping. So this is kind of split up into two segments. We have first algorithm development, and so what we're going to do here is to optimize methods for robust instant segmentation of 5 cell classes. This is just all 5 cell classes probed. I'll go through the data set in just a second. And so we're going to train and validate these models and then evaluate on an independent test set to make sure this will generalize to our full cohort. So our data set, we have 55 lupus nephritis patients, 36 were ESRD negative, so did not progress to renal failure, and 19 of our patients did progress to renal failure. And this is within a 2-year follow-up. So on kidney biopsies from these patients, we did 6-channel fluorescence confocal imaging. And we collected discrete image ROIs in regions of lymphocytic infiltration, since we were specifically interested in the role of lymphocytes within these pathologies. We also looked at plasmacytoid dendritic cells, also known as PDCs, myeloid dendritic cells, which I'll refer to as MDCs, and the 3 lymphocyte populations that we were able to probe, given the 6-marker limitation, were B cells, and then 2 categories of T cells. We had our pan CD3 marker for T cells, and then we also stained for CD4, so we were able to discern helper T cells from the remaining populations of T cells. So of our 55 patients, we selected a random subset of images from 31 patients for a training set of 342 images, or training, this is, sorry, not just the training set, this is all of our manual segmentation, so training, validation, and testing. And this resulted in about over 10,000 cells that were hand segmented by our experts for this task. And so you can see an example of the ground truth for a single image ROI here in the RGB mask image. See if you can see my mouse. And this image right here is an example of the ground truth that was provided by the human. And so once we look at the distribution of ground truth, we notice that the colors are matched here to the text. So our lymphocytes, B cells, CD4 T cells, and CD4 negative T cells make up about 85% of the cell populations within these images, while MDCs and PDCs only make up about 15%. And in this provides a class imbalance issue, which needs to be mitigated to accurately detect all cell classes. So you want to have as balanced cell classes as possible for these algorithms, which when you're looking at lupus nephritis in these regions of dense lymphocytic infiltration, there is an inherent class imbalance in the biology. So we can't exactly augment by providing DC specific images because there will be lymphocytes in those images as well. So what we did is we trained Categorical Mask R-CNN, and so we split our 6-channel images into 2 sets of images. So on the top, we have our Mask R-CNN that's trained specifically to segment 3 classes of lymphocytes. So we have our CD20 to identify our B cells, and CD3 and CD4 to identify our 2 classes of T cells. We also have DAPI within each image stack just to confirm that there is a cell there, and that's for the ground truth, as well as the network uses that DAPI. And then we also have our dendritic cell arm. So we have our BDCA2 to stain our PDCs, our CD11C to stain our MDCs, and then DAPI for our confirmation that that is a cell body. After training each of these algorithms, we predict and then combine the predictions to get our 5-class instant segmented image on the right. So now we want to look at performance on an independent test set. So these patients are unique to the patients that were in the training and validation set. And so you can see 2 examples here of the ground truth provided by the human and the predictions provided by the network. I've kind of strategically picked 2 images, one that's less dense and one that is very dense with lymphocytic infiltration, because most traditional computer vision methods are not great at detecting cells in crowded regions. But you can see here that the network does a pretty good job of detecting these cells in dense regions of infiltration. So the first metric that we want to look at is recall, which is specifically how well do we detect what is present. It's analogous to sensitivity. And so we do a pretty good job with our lymphocytes and our PDCs. Our MDCs are not as great. We kind of went back later. There is some channel bleed through with the CD4. So this is a panel that we maybe wouldn't use in the future, but pretty good performance on our lymphocytes and our PDCs. Then we move to precision. So how well do we avoid false positives in our object detection class? And again, we have pretty good precision on our CD4 positive and our B cells and our PDCs, not as great precision on MDCs, and CD4 negative T cells is getting there, but there are some false positives labeled that were actually CD4s. And finally, intersection over union was pretty consistent over all cell classes. So that's how well do our shapes match. And given the differences between a person doing this, the same person doing it over and over again, these IOUs are pretty standard. So now that we have developed this algorithm and characterized its performance with regard to our lymphocytes and DCs, and we know how well we can detect each of those cell types, we want to deploy this algorithm to actually answer biological questions. So we use our trained models to make cell predictions on the full lupus nephritis data set, and that includes the training and testing data, as well as all of the patient images that were not manually truthed by a human. So again, our full lupus nephritis cohort was 55 patients, and that was about 1,250 images overall. So the models were developed for instant segmentation, which is cell detection, segmentation, and classification. The manual truth was 342 images, which took humans about 150 hours to do, and that was only about 10,000 cells. When we deploy the model, after testing. In less than an hour, the model was able to detect over 120,000 cells within this data set. So rapid detection of cells, not a task that a person could do. So now that we've detected these cells in this data set, what can we learn from this spatial data? So first, what we want to do is extract simple cell frequency features. And this is something that we could do with a different modality, something that's not spatial. But the fact that we do have image ROIs gives us a proxy for density. So we can calculate local cell density. And if we look at B cell density, we notice that high density B cell regions are associated with ESRD negative. So we're expecting those to be patients that are responding to current standard of therapy for lupus nephritis. Additionally, our ESRD positive group, we found an interesting cohort which we're calling ESRD current. And so we had a subset of 5 patients that progressed to renal failure within 2 weeks. Some of them actually presented in renal failure. And so if we look there, we see that there's a forward trend of essentially the severity of disease. So high regions of dense B cells are associated with ESRD, and our ESRD current patients have the lowest dense regions of B cells. And so conversely, when we look at our CD4 negative T cell population, those dense regions of CD4 negative T cells are actually associated with renal failure, which is interesting because this is actually our least characterized class. They're essentially characterized by an exclusion. And so we see the same trend that we did, or I guess the inverse trend is what we did with the B cell density. And so now we can assume that our CD4 negative T cells, dense regions of those cells found in lupus nephritis, is associated with patients who do not respond to the current standard of care. So now that we have done these cell density calculations, we can get more detailed on what we do with the spatial analysis, do a few more interesting things. So we have implemented something called dbScan, which is a density-based clustering algorithm. So dbScan was actually developed to analyze data in the feature space, but we kind of flipped it on its head and decided we were going to use it in the physical space to detect neighborhoods of cells. And so you can see the different neighborhoods of cells outlined in this image ROI over here. Once we've detected those cells, we can now extract features from the cell neighborhoods. So we have have features that are related to cell frequencies. So how often specific classes of cells pop up in an aggregate, the proportions of those cells, the neighborhood shape features, frequency ratios, and finally morphological features of the cells themselves within that neighborhood. So then we want to optimize the number of groups, since we want to categorize different types of neighborhoods and see if different types of neighborhoods are associated with progression to failure as well. So once we categorize those neighborhoods, we found that 6 neighborhoods was the optimal number of groups using some k-means clustering algorithms. And so we have 6 unique types of clusters that come out of that, mostly associated with specific cell classes. And what we find is that just like the density analysis, the CD4 negative T cell neighborhoods, which are found kind of within the ROI level, are associated with patient outcomes. So CD4 negative neighborhoods, the more prevalent they are within a patient, the more likely that patient is to progress to renal failure. So with this lupus nephritis outcomes cohort, we have found that computer vision methods facilitated cell quantification in these immunofluorescence images. Simple local cell density measurements reveal the association between B cells, CD4 negative T cells, and patient outcomes. Specifically, high local densities of B cells were associated with ESRD negative patients, and high local densities of CD4 negative T cells are associated with ESRD positive patients. Additionally, more advanced spatial analyses revealed that there were outcome associated neighborhoods of cells, and that was those CD4 negative T cells. And with that, I would like to stop here and see if there's any questions so far. If there aren't any questions, I can go ahead and move on. Oh, Dr. Dork. Oh, you're muted if you're talking.
Oh, I couldn't unmute myself a second ago. The Mask R-CNN, you adapted that for this application, is that right?
Correct, yes.
And is that something you invented? Did you invent that?
No, this was something that came out of Google in 2016 or 2017.
Okay.
But it's kind of shown to be the state of the art for instant segmentation in natural images. And deep learning moves really fast, and it's still pretty much state of the art. There are a few other instant segmentation algorithms that have popped up. Some might be have less parameters than Mask R-CNN. Others might be a little bit faster, but in general, Mask R-CNN still produces the best performance.
With cancer, the shape of the cells is very important. Have you looked into quantifying distributions of shapes of cells at all?
Yeah, we have actually. So not necessarily related to outcome. We did extract those features and we didn't find anything associated with outcome for lupus. But we did an interesting analysis about fixation method of the tissue and how that affects the different cell shapes. So like fresh frozen tissue versus FFPE tissue, you would end up with different results basically, depending on which of those fixation methods you have of your tissue.
Thank you.
Thanks. All right, Rachel.
Hi, I'm interested in kind of some of the trouble that you had with the CD11C positive cells. Like, you know, when segmenting myeloid cells, a lot of times because of their morphology, they don't necessarily have a nucleus that's in the same plane, or, you know, that nucleus might not be in the center of the cell. Are part of the problems that you had related to kind of the localization of the nucleus, and are there ways to kind of get around that issue in the segmentation?
Yeah, absolutely. So I think, I mean, you're spot on. The DCs do like reach in and out of plane. And so you can see positive signal in a slice that's maybe not where the nucleus is. And I think a lot of the issues with the DC population is actually the consistency of the human viewer. So there's some really clear positive signal in the CD11C channel, but then you can't necessarily relate that back to a nucleus. And so if the person establishing ground truth, sometimes they relate it back to a nucleus, sometimes they don't. There's some variability there. And so that means that your ground truth is essentially corrupted. So there's some difficulty there, and there's also bleed through with the way this panel was set up between the CD4 channel and the CD11C channel. So you have some false positive signal in there that bleeds through from the CD4 as well. So it kind of comes down to two factors, which is panel design and the morphology of the CD11C and establishing the ground truth for these supervised learning algorithms.
Thanks.
You're welcome. Thanks for that question. All right, I'm going to take the two more questions that I see and then move on. Toby?
There we go. I have a couple of questions. So what was your sampling? So these look like ROIs, presumably. So what was your sampling strategy for this?
So this was this data set was originally taken to look at cognate interactions between T cells and dendritic cells and B cells, actually. So they were specifically focused on regions of lymphocytic infiltration. And I'll talk a little bit later. Once we realized that we had outcomes for this and could do these interesting analyses, we're like, we need to be taking whole slide images. So then later on, I'll talk about moving to data sets like that and how much more powerful it is as opposed to these discrete ROIs. But it was nice that the discrete ROIs had that built-in proxy for density. It made those calculations really easy.
Well, my other question was, have you applied it to an independent data set, but maybe you're getting to that at some point.
This specific, so we did generalize this to RA and rheumatoid arthritis and a mixed rejection of renal allograft, so kidney transplant tissue. And so it generalized pretty well, specifically the lymphocyte network. That was much better because the baseline performance was so much better. I don't have that data here, but it's published in an SPIE proceeding, and it works pretty well as long as the same panel is used. If the panel is different and there's like different tissue autofluorescence from the different laser lines, the performance goes down. Thanks for that question. And I see Greg is the name, but I think that's yeah.
Sorry, we are.
A little bit of an echo, yeah.
So basically we are grouped in the chat. Can you hear me now?
Yes, that's much better.
Can you hear me now?
Yes.
But we couldn't hear.
Since we're hearing from here.
So we're using a screen.
They can hear us.
You can hear us?
Yes.
Oh, okay. So basically, I just ask. So that's cool. I like the idea is basically you define the neighborhood based on the spatial information. So, but basically, have you thought about basically if you analyze the gene expression data, right? We can also define the neighborhood based on transcriptomic variation, right? So do you have any insights if you combine or integrate the spatial transcriptomic data, how you merge the variation from the neighborhood, like similarities between the gene expression level and the spatial level?
Yeah. So that's something I've been kind of interested in. NanoString has their cosmic system, which came out, so you can do proteomic imaging and spatial transcriptomics with that, and so you have that direct comparison that you can do. I don't know if there has been any studies that have like done spatial transcriptomics and a proteomics thing on the same samples, but it's definitely something I'm interested in, and that's like what the lab that I work with at UChicago is moving forward with the cosmic system from NanoString. I think it's a really interesting thing is analyzing the data across those, seeing how they relate and how they differ. But I wouldn't know how to merge that information yet because I don't know the distributions of those two data sets, but I think there's a lot of work to be done in that space, and it's really exciting. All right, so that was great. Thank you guys so much for all those questions. You guys can still see my slides. I haven't changed that at all. So I'm gonna go ahead and move on to the next section. So there are pretty clear limitations with this data set, which we've addressed already with some of your questions. First is the discrete images capture only a subset of a section of a sample of diseased tissue. So there's a lot of sampling bias happening in this data set. Second is the standard immunofluorescence imaging limits us to about 5 underdefined cell classes. Specifically, what are these CD4 negative T cells? We normally assume that CD4 negative means CD8 positive, but is that the case in this data set? So if we look at our multiplexed immunofluorescence microscopy, as I said earlier, we're limited by the physics of the imaging. So if I have my DAPI channel, my 405 spectra right here, and then I collect a few more stains, you can see if I were to go any deeper in here, there would be a lot more channel bleed through. So the spectra of these specific fluorophores limit the number of channels that I can acquire in a specific field of view. So this limitation is known as the color barrier of fluorescence microscopy, and this barrier significance is what significantly limits the number of phenotypes probed in a given sample or image. So a few methods have come out in the past 5 to 10 years that break the color barrier. And these are referred to as highly multiplexed immunofluorescence microscopy methods. First, we have tissue cyclic immunofluorescence. And what they do here is they lay down a layer of antibodies, 3 to 4 channels, or primary and then secondary. They bleach the fluorophores on their secondary antibodies and then re-image again. So as you're bleaching, you're depositing a lot of energy into the tissue, and the tissue gets destroyed pretty quickly here. So that limits the number of markers that you can acquire. Next is MEVI. So this is, instead of fluorescence imaging as we typically think about it, you have an ion beam that knocks off a heavy metal or some other particle that's on your antigen, and then you send that through mass spec and map that back spatially. It's an expensive system, provides really great images, but it's hard to generalize to. And finally, we have Codex, which you lay down your primary antibodies. These have oligos attached to them, and there's a series of reporter molecules that have the complementary oligos. And those are really easy to break chemically without destroying the tissue. So you can do this cyclic imaging while maintaining the integrity of the tissue. And so we have validated a strip and reprobe that's similar to Codex. We've since bought a Codex after doing this validation cohort. But now we have a highly multiplexed immunofluorescence data set of lupus nephritis. So the previous data set had outcomes associated with each patient. This 18 patient cohort, we don't have 2 years of follow-up data for these patients, so we don't have those outcomes, but we can use some of our findings from the outcomes cohort to learn more. So this cyclic strip and reprobe we used to achieve a 25 marker panel through 8 cycles of imaging. And in this data set, we have cell membrane markers, nuclear markers, and contextual markers. And today I'm just going to talk about what we've done for detecting lymphocytes within this tissue. And as I mentioned earlier, these are whole site images. You can see an example of the full biopsy on the left. So this image is actually about 40,000 by 40,000 pixels and 25 channels deep. So this is 6 channels that I've selected to show of some interesting lymphocyte markers, but you can tell that there's a lot of interesting information here to mine. Oh, keep going backwards. So I think this group knows a pretty decent amount of about lymphocytes and immune cell populations, but just a quick primer. So, and this also talks about what we have in this data set. First, we have our pan T cell marker to look at our T lymphocytes, which is our CD3 marker. And from there, we have our kind of umbrella populations of T cells. We have CD4 to mark helper T cells, and those can be further categorized as ICOS positive or negative, FOXP3 positive or negative, PD1 positive or negative. There are a few more markers in there, but I won't go into all of them. We also have our CD8, our cytotoxic T cells, which can also be further categorized by other markers. And then we have our double negative T cells, which express neither CD4 nor CD8. We also are going to look at, for this presentation, our B cells, which we've marked in this data set with CD20, and our plasma cells, which we've identified with CD138. So our first step in analyzing these images, after pre-processing all the stitching and alignment of these highly multiplexed images, is to find these inflamed regions. So basically, we want to minimize the computational effort kind of put into the instant segmentation, as that's a pretty computational heavy task. So we have found the inflamed regions. We built a data set of 512 by 512 tiles after breaking up these large images. And this training set is composed of inflamed and uninflamed regions. So the inflamed regions either have positive CD20 signal or positive CD3 signal. So we're just looking for areas of lymphocytic infiltration again, and rejecting all the scarred tissue, basically. And so what we get is, so we've trained a VGG19, sorry about that, a VGG19 to do this classification automatically for us. And so we had about 500 total images to train this, and we're able to get an AUC of 0.99 to filter those images out and reject the scarred tubules. From there, we move on to our Mask R-CNN fine-tuning. So rather than training Mask R-CNN from scratch, as that takes a lot of training data, we want to minimize the need for human intervention in here. So our previous high-resolution data set which I talked about, 512 tiles to match the image size that we've broken up the high dimensional set into, we end up with about 1,400 image tiles from this data set, and we can leverage the ground truth there to pre-train algorithms. So now if we look at our highly multiplex data set, we have a larger pixel size, so we're going to have to learn the difference between these 2 pixel sizes, going to a lower resolution images. And then, as I mentioned, we have whole slide images that we've broken up into 512 tiles, and we want to minimize the need for ground truth. So what we've done is we've initialized 2 separate networks. So now we have a B cell based Mask R-CNN, and so we've pre-trained this with the B cell images from the high resolution data set, and that training set was about 1,200 tiles. And then we only needed about 100 images to fine tune this Mask R-CNN. So rather than doing another 1,512 by 512 image truthing. We only need to do 100 images of ground truth, and then predicted on both CD20, so our B cells, and CD138, so our plasma cells with this network. We also have our T cell network over on the right. And so we had our 1200 image tiles from our previous data set, which we initialized Mask R-CNN with, and then we're able to fine tune with about 100 images. The important thing to note here is that while our pre-training data, our initialization data had CD3 and CD4, our fine-tuning data, we can change out that second channel. So we still want to use CD3 as entry criteria to define our T cells, but we can change out CD4 for CD8, ICOS, PD1, and other T cell markers. So this, this network can detect a number of T cell markers to help define that specific class of T cell. Oh. And so after testing on an independent set of patients on plasma and B cells, the single channel network had an F1 score of 0.82 on those two cell types, and across several T cell types, the T cell network had a score of 0.88. So we're detecting these categories of cells pretty well in this high dimensional data set. So once we've found the lymphocytes within this tissue, we can do a similar analysis where we do our DB scan to do our spatial clustering and identify these cellular neighborhoods. And then we can do our neighborhood characterization and classify these neighborhoods based on the residence, morphologies, and the intercellular distances. And so these neighborhood features extracted from the highly multiplexed data set can be used to characterize the detected neighborhoods into the outcome associated bins from the last data set. So we know that those CD4 negative neighborhoods were associated with patient outcomes. Can we find those neighborhoods in this data set, and what's going on with those neighborhoods? We want to know more about them. So first, we just characterize our more granular phenotypes of lymphocytes. And so we have our T, B, and plasma cells pulled across all samples. And so if we look at the distribution of what would be our CD4 negative T cells, so our CD8s and our double negative, you can see there's actually pretty even representation of CD8 positive T cells and double negative T cells in these images, and a pretty significant number of plasma cells as well. On the right, you can see further classification based off of PD1, ICOS, and FOXP3. We have our CD8 exhausted T cells, defined in the light blue in the second column. And we also have T regulatory and T follicular helper cells highlighted in the CD4 positive column in those different shades of red. So we can identify more specific types of T cells within these images as well. So next we move on to the regional and structural segmentation. So we also want to know where these networks are relative to the structures that that they might be destroying. So if these infiltrates are responsible for destroying kidney function, what are they destroying, and where do the dangerous neighborhoods reside? So first, we just did a full biopsy segmentation using a conventional Gaussian filter and threshold to get the full biopsy segmentation. There aren't a lot of glomeruli in these images, so we just did manual segmentation. If we had a large enough data set, we could build up something to do automatic segmentation, but with 1 or 2 per image, hand segmentation was the way to go. And finally, we did automatic tubule segmentation by training in Mask R-CNN on the DAPI tiles. Again, those tubules were hand segmented, and that Mask R-CNN at the pixel level resulted in an AUC of 0.9. And so once we have those 3 structural segmentations done, we can merge with the automatic cell segmentation to get the image on the right, where we have all the information about where the cells are relative to the structures in the kidney. So if we look at the regional segmentation, we can see that tubules make up about 40% of the biopsy area, glomeruli around 5%, and the tubulo-interstitial space makes up the remaining percentage. So if we look at the neighborhoods that we found, so we're specifically looking at the CD4 negative neighborhoods, which are highlighted in cyan in this image. Those were associated with ESRD positive patients. We also have our BT neighborhoods, which our collaborator in rheumatology wanted to look into as potential tertiary lymphoid-like structures, and we wanted to further characterize those as well. And so what we find is that these large BT neighborhoods are often surrounding the glomeruli. And so they're significantly closer to the glomeruli than the CD4 negative neighborhoods, as well as all other detected neighborhoods. And the converse is true with the distance to tubules. So the BT neighborhoods are farther from tubules than the CD4 negative neighborhoods. So those outcome associated neighborhoods are really lying within that tubulo-interstitial space. So when we look at this multiplex cohort, we've used a series of deep learning models to classify image tiles, detect lymphocytes, and segment structures in this data set. More specific cell phenotypes were identified within these images while maintaining spatial relationships. The CD4 negative T cell compartment included both CD8 and double negative T cells. The CD4 negative T cell neighborhoods, which were previously associated with ESRD positive outcomes, were found to be in the tubulo-interstitial space and not near glomeruli. And with that, I'm going to stop for a brief few questions before I move on, if anyone has any questions on this section.
Do you think that the double negative T cells are gamma delta T cells? Did you do vaccines to identify gamma deltas?
Those are like mostly gamma deltas. So that's spot on. We did a separate staining on just like a small cohort. I think it was 8 patients where we just stained for CD3, CD4, CD8, gamma delta, and DAPI. And most of what we could find in the double negative population were gamma delta positive. We haven't done like a direct computer associated or a computer assisted quantification of that. That was just done through manual because there's just a handful of images, but definitely gamma delta.
You have an idea yet of whether the CD8 positive or the gamma deltas are, really the bad guys in this?
We do not know yet, no.
So I have a quick question. So yeah, the TB neighborhood looks really interesting. So do you know how? But because actually for the inflammation in the tissue, the fibroblasts also really involved in driving the tissue inflammation, right? Called like tissue priming. So do you think how you can capture the mesenchymal immune interactions in the colocalization using the imaging? Because now we can only do a limited, right, features, right? So how you can detect more such, neighborhoods colocalization related with tissue inflammation?
Yeah, so this is just, I think this is maybe like 10 or 11 out of the 25 markers that we have. Fibroblasts aren't in this panel. And so we have another experiment getting started where we actually have a 40 marker panel and can explore in even more granular detail all of these relationships. But what we're interested in moving forward to is like looking at the dendritic cells and like the antigen presentation within these neighborhoods and seeing if we can detect antigen presentation within these large BT neighborhoods. And so that's what's driving the research behind those interesting ones right now. Great. All right, I'll go ahead and move on. Try to zip through the rest of this pretty quick. Thank you guys so much. These questions are fantastic. Oh yes, there's that comment on the large BT neighborhoods. Next, we want to know how generalizable these methods are. So our second clinical motivation is triple negative breast cancer. It's an aggressive type of breast cancer that's defined by a lack of hormone and HER2 receptors. It has a greater risk for recurrence than other subtypes of breast cancer. Out of the 1 million cases diagnosed annually, about 20% of them are breast cancer. The risk factors are genetic. If you have a BRCA1 mutation, you're much more likely to get TNBC, ethnicity, and age. So unfortunately, young Black women are much more likely to suffer from triple negative breast cancer than their white counterparts. Additionally, after we correct for this prevalence issue, Black women are still have a lower 5-year survival rate than their white counterparts. So we want to investigate TNBC a little bit more. and specifically the tumor microenvironment, tumor-infiltrating lymphocytes are generally associated with good patient outcomes in TNBC. If we look at just a typical H&E pathology image, we can identify regions of tumors and regions of lymphocytes and study here, but we want to know a little bit more about lymphocytes and the different compartments of lymphocytes that exist here. Additionally, the spatial distribution of tumor-infiltrating lymphocytes varies, and this is associated with outcome as well. So if you have a cold tumor or very few lymphocytes within that tumor, you have the worst prognosis. But if you have a mixed organization of your lymphocytes in your tumor relative to a compartmentalized organization, that is a difference in prognosis as well. So the thoughts are that this compartmentalized immunity could be tertiary lymphoid structures, which might also be associated with good prognosis. However, there's a need to better understand the spatial immunity in TNBC with a higher phenotypic resolution than can be provided by H&E staining or conventional confocal microscopy. So we have a second data set, a second highly multiplexed data set, I should say, of 19 TNBC core needle biopsies. We've used the similar staining method, but a different pathology requires a different panel. I've shown an example of one of the TNBC images here on the right, with just a few of the stains highlighted. Specifically, you can see the tumor in green and a few of the lymphocyte markers as well. So we want to see if we can leverage the ground truth from our lupus data sets to accurately predict cells here. So we have a series of fine-tuning experiments that we've done. First is to fine-tune with the matched resolution lupus, because the TNBC images have the same resolution as the highly multiplexed lupus data set. Second is to fine-tune with just TNBC. And third, to leverage all the ground truth we have, is to fine-tune with the merged matched resolution lupus and TNBC data sets. So if we look at the overall performance of these fine-tuning methods, so if we look at our data set, or our Mask R-CNN trained on our initial lupus data set, that high resolution data, our recall is pretty low. And so any amount of fine-tuning improves our recall. Still not to a great level, but significantly over the initial trained network. However, precision is still very low. There's no significant decrease, but also no significant increase. There are a lot of false positives here, but we still have pretty good IOUs across all of this. And so combining lupus and the triple negative breast cancer data sets does improve sensitivity when you're fine tuning, but we have a lot of false positives, particularly in the CD4 negative T cell population, and that's due to some issues with the channel in the CD3 positive population. So if this doesn't generalize, what is the best way? So if we're looking at instant segmentation with these higher dimensional images, so these are just 10 channels from one ROI of a highly multiplex data set. And you can imagine, like, defining ground truth for all of these is pretty difficult. So supervised learning requires a substantial amount of manual truth to perform well. And also humans are not consistent at this task. So if we look at class level instant segmentation done compared human to human, so we have one expert done in red, white is where the two readers agree. Red is where reader A has defined a cell, but not reader B. And yellow is where reader B has defined a cell, but not reader A. And so if we look at the F1 score precision and recall between humans, we're very low, pretty low recall of 0.67 between two readers. So theoretically, stain level instant segmentation would be easier, just defining, okay, if this cell is positive for this stain, we're going to segment it rather than doing a multi-stain classification. Humans are also not great at that task, still a low recall, about 0.65, and even a lower F1 score between readers. So what we want to do now is just kind of break open this pipeline. It's going to be difficult for these high channel data sets to be able to define the ground truth that we need for true supervised learning at the full instant segmentation level. So if we look at these images, what's the simplest task that will be most reproducible across humans? And that's probably going to be object detection on our entry criteria for a cell, which is DAPI. And so what we've done is we've trained YOLO, which is an object detection convolutional neural network, just to detect DAPI in these images. So YOLO is a very small network compared to Mask R-CNN. It needs a lot less training data and it requires a lot less training time. So we've trained a YOLO 5 network on detection of triple negative breast cancer DAPI, so specifically the nuclei. And right off the bat, we have an F1 score of 0.89, which is pretty high. And so that's on a test set of TNBC images. If we directly apply that to lupus images with no fine tuning whatsoever, so no extra truth required, we have an F1 score of 0.83. So we're already in the realm of improved performance just by changing to a simpler task where we have more consistent ground truth. So if we look at this, object detection in lupus and compare the YOLO results to 5 human readers, there's no significant difference between the YOLO versus the human reader and the human reader compared to another human reader. So if we break this down to the sample level, we can see that Mask R-CNN is just as consistent as the reader within the readers at the sample level as well. So we can also look at the sample level comparing between readers. And so we have a series of studies going on right now to ensure that this is actually the best, most robust way to define ground truth for these types of images. So once we have done this DAPI detection, so we can pull the patches from each channel associated with this cell. And what we want to do here is for each channel, we can calculate cell features or bounding box features. So we have size features to calculate, just image intensity, texture features, and eventually we want to move to supervised and unsupervised deep learning for classification of these different channels. So what this allows us to do is to move to kind of a little bit more of a flow cytometry approach where you can look at the differential expression of markers for a given nucleus that was detected. What we're working on right now is the normalization here. So we have normalization across samples to deal with. As you can imagine, there's different uptake of antibodies across patients. And there's also different tissue autofluorescence associated with each patient. Another issue is that there is a spectral variability to that tissue autofluorescence, and so that affects the correlation of markers expressed on a given cell as well. And really quick, before I finish up, I want to talk about some future directions. Where do we want to go with all this? So moving forward, I still want to keep mining these quantitative spatial data from human tissue. Combining all these, I think there's a wealth of data in archive biopsies that we can access through computer vision. And I think we're just really scratching the surface of what we can learn from archived human tissue. I'm also very interested in merging multi-scale patient data. I have a project sitting on my desk right now where I have matched MRI data with some lumpectomy sections actually from triple negative breast cancer patients, and merging those multi-scale imaging features I think is going to be really exciting in terms of learning more about what you can figure out about patient prognosis from these earlier images. Also, doing cross-modality synthesis, so going from one domain of images to another, I think will allow us to learn more about real-time feedback for endoscopy. And finally, something I'm really excited about is the interpretability and generalizability of these algorithms. Traditionally, people will use something like you see in this image called GradCam, where you highlight where the network is focusing on. But we have these classification algorithms that work really well, but we don't know why. And I think kind of by tying in some of the features that we can learn from instant segmentation and spatial mapping, we can generate some knowledge about some human interpretable features that are associated with the network's decisions on classification. And with that, I'd like to thank you all for listening, and thank you all for the great questions so far, and everyone who has worked in these projects at the University of Chicago. And with that, I will take any more questions that you guys have.
It is very challenging to not be able to hear all the claps that you're getting with little emojis across the board. Thank you so much, Dr. Durkee. Do we have any more questions? We have about 2.5 more minutes.
I'll ask one on the explainability front. So you were talking about kind of how you might attribute predictions, given that you have sort of some additional information about sort of cell types that are there and potentially markers on those cell types. Are you thinking about like attribution as potentially including like some sort of almost semantic description of what might be different in a certain setting, or are you really thinking about it as kind of like, how are you thinking about explanation, maybe is what I'll say? Okay,
so mostly like people will take the activation layers and see what's being activated using like a GradCam software. But you can also do like correlations between like conventional features or human interpretable features and those features, the deep learned features that are learned by the computer. And so what hasn't been done yet is, so we have like a network that is really good at predicting whether or not a patient will respond well to like neoadjuvant therapy or something in breast cancer, right? And so we don't know exactly why that works, and it would be really helpful to know why a patient will respond or won't respond, because then we can maybe think about like, well, what's different about these patients that don't respond in their cellular environments. And so once we like map all the cells, we can then develop human interpretable features about like the spatial layout of these cells and see if that's associated with what these networks are finding in the classification task. And so it really is kind of using deep learning to investigate deep learning, but really on like the cell detection level rather than the patient level classification.
Thank you very much again, Dr. Durkee. I think with that, we'll move on, and thank you everybody for joining seminar today.
Yes, thank you so much. You guys had such wonderful questions.
Cheers. Goodbye.